This is Elyse Hope using experimental evolution to engineer a low flocculation yeast strain.
Good evening, everyone, and thank you for the organizers to invite me to speak in this session. And I'm going to start a conversation right now about experimental evolution that I hope to continue with many of you throughout the week. And in the Genome lab, we use experimental evolution to see how yeast adapts to selective pressures, but it's also a useful tool for engineering. If we have a strain of yeast, or many strains of yeast that frequently evolve traits that are good for them but may be bad for us, traits that we wish they would not do and would like to explore engineering as a tool to eliminate them, we can do the evolution experiments many times and ask some interesting questions, including whether or not they all take the same adaptive route, and also whether we can reduce the potential for the strains to evolve the trait in the first place, which is an engineering question. And our favorite tool in the Genome lab for experimental evolution is the continuous culture chemostat. And advantages of this system are that it maintains a constant growth rate and also constant nutrient limitation throughout the experiment. We can observe evolution in real time if we sequence throughout the experiment, and it's also high throughput. So the experiments that we do, typically we'll do 32 or 64 in parallel, but we actually have the capacity to do up to 128. And during the course of an experiment, the yeast are evolving in response to the selective pressure, but they're also evolving in response to the physical constraints of the environment. So as a result, continuous culture is actually a good mechanism for studying aggregation. If we start with an ancestral strain that has a loss-of-function mutation in a transcriptional regulator of flocculation, which is cell-cell adhesion, and we evolve that strain for 300 generations, and we do that 96 times, we see about 35% of the time one or both of these traits evolve. Wall sticking, which is adhesion to the glass vessel itself, and also aggregation, this clumping phenotype in liquid culture. And this is a trait that's good for the yeast because it increases their residence time in the culture and decreases the likelihood that they'll be diluted out. But it's bad for us because a lot of the experiments that we like to do require all of the cells in the culture to have equal access to the limited nutrient. So this is an example of a trait that is good for them but bad for us. Similarly, outside of the lab, this trait allows yeast to persist on hospital surfaces and even to more successfully colonize the human body, especially following antimicrobial treatment, which this flocculation behavior allows it to resist. And also in industrial settings, any sort of industrial fermentation, if, for example, biofuels production, this trait is a nuisance there as well. So it's clear that there would be a lot of advantages to not only better understanding the genetics of this trait, but also to develop some engineering routes where we might reduce the likelihood that it evolves in the first place. So to this end, we selected 23 clones for study from these evolution experiments. These all had the aggregation phenotype, and we asked, how are they doing it? So if we get into the sequence information for these clones, we can ask what genes are contributing to aggregation in the first place, whether we can identify any new contributing genes at all, or if it's just the usual suspects previously identified in the literature, and also whether there's one favored adaptive route or not, because that determines the engineering potential for this trait. If all of the strains acquire this trait in completely different ways, then we would have to work really hard to engineer a strain that doesn't do it at all. We know from our initial sequencing that all of these clones harbor many mutations, and so in order to get right at causality, we did bulk segregant analysis. And the premise here is that if we have an evolved strain with a phenotype of interest, I'll show here with the mouse on the right, and the phenotype in this case is a settling in liquid culture, and we backcross that clone to the ancestral strain that does not have that phenotype, then we can look at how the phenotype segregates in the meiotic progeny. So we're looking for traits, or we're looking for examples of segregants that have this trait segregating 2 to 2. So 2 of the segregants in the tetrad have the settling trait and 2 of them do not. And that would be indicative of a single causal mutation rather than a more complex trait. And then we pool the segregants into pools depending on the phenotype. So we have one pool with the settling phenotype and the other without, and we look for mutations that co-segregate perfectly with that trait. And we did observe this 2-to-2 segregation, it's worth mentioning, in all but one of our clones. So this was a very fruitful avenue of exploration for us. So if we follow this into the results and we look at just one single clone, this is one evolved clone, and it had 4 candidate mutations in our initial whole genome sequencing, which are shown here, starting with ROX3. And you can see that if we look at the percentage of the mutant allele represented in that pool with the phenotype, we can see that only ROX3 perfectly co-segregates with this trait, and we would call that as the causal mutation in this case. So if we combine all of the data that we've acquired for these strains so far, I started preparing this talk with 2 unsolved of our 23 clones. I can actually say that within the last half hour, we have 1 unsolved, thanks to the hard work of one of my co-authors. And the most common adaptive route by far was this transposable element mutation in the promoter of flocculin gene Flo1. We saw that in 12 of our clones. Also 4, now 5, loss of function mutations in the gene TUP1, a known repressor, and 2 mutations in ACE2, which is a transcription factor involved in mother-daughter separation. So those were actually not strains that exhibited flocculation, but rather a mother-daughter separation defect. ROX3, as I mentioned, and a smattering of other mutations, and now only 1 unsolved, which we are working hard on as we speak. And the primary story that really emerged for us at this point, driven in large part by those transposable elements in front of Flo1, is a common route to flocculation through Flo1 regulation. So Flo1 codes for a cell surface flocculin protein. It has a well-documented role in the literature in cell-cell adhesion that's reversible, which is our working definition of flocculation. And so it's understandable why a beneficial route to flocculating would be to increase expression of this particular gene. One way to increase expression is to lose a repressor. So TUP1 is a known repressor of Flo1. We observed mutations in TUP1, loss of function mutations in TUP1 in 5 of our clones. So that's a valuable route to express that gene. Also, ROX3 is a component of the RNA polymerase II mediator complex, which is also a repressor of Flo1. Although ROX3 has not been previously linked to flocculation, ours is the first study to do that. There are other components of that complex where loss of function mutations in those genes have been linked to flocculation. So it's a reasonable hypothesis for what's going on there. Another way to increase expression of Flo1 would be to activate it with the insertion of a transposable element. So as I mentioned, we see this in the majority of our clones. These purple lines coming from the transposable element indicate the 2 hotspot areas where we saw these insertions in the promoter. And in all cases, we had a full-length insertion in the reverse orientation with respect to the gene, which is the most common conformation for an activating arrangement. And there's a long history in the field of transposable elements frequently modifying gene expression in yeast, actually starting as early as 1980 with our esteemed speaker from earlier this evening, Rothstein, who found an overexpression mutant of iso2 cytochrome C that turned out to be due to a transposable element insertion in the promoter. And studies onwards from that and into very recent papers have tried to identify which components of the transposable element are responsible for this activating behavior. So now that we know a little bit about the genetics of this trait, we can finally ask if we can engineer a strain that no longer evolves flocculation, or it takes longer to evolve flocculation. So we started with these 23 aggregating clones, and we identified a common route through Flo1 regulation by which they are acquiring this flocculation trait. And I just want to point out at this point that this could have gone very differently. All 23 of these clones could have acquired this trait in very different ways, leaving us with a difficult engineering problem. But because of the particular result we found, this common route to Flo1 regulation, that enables us to make a very intelligent engineering choice and ask if, by simply deleting this gene, if it will take longer for the strains to evolve flocculation, which is exactly what we did. So if we continue around our cycle, we evolved 32 wild-type strains in comparison to 32 of this Flo1 knockout strain in our miniature chemostats for over 250 generations. And we asked during that time how many of the wild-type and how many of the knockout strain evolved the flocculation trait. And we found that 8 of our wild-type strains evolved the trait, but only one of our knockout strains evolved flocculation. So at this point, we were able to say that deleting Flo1 did significantly reduce flocculation in our experiments, and this is now the favored strain for evolution experiments in our lab. And if we follow up with that one knockout that flocculated, we find that this escaping strain has a TY insertion in the Flo9 promoter, which is another of the flocculin genes. So you can imagine that we could continue with this trait, either engineering with respect to top1 or perhaps going after the TY mechanism itself or Flo9. But we can also use this general approach to go after other traits that we might want to reduce. So I talked earlier about that wall sticking trait that we see very commonly in the chemostats, also the separation defect that we observed in 2 of our clones. And neither of these were significantly affected by the deletion of Flo1, which we would expect based on our understanding of how these traits occur. But if we sequence some of the clones from some of the strains that evolve these traits, we find that ACE2 loss of function mutations can actually cause both traits. So a very reasonable route for engineering would be to increase the copy number of ACE2, either by using a diploid strain or by engineering or maybe fusing it to an essential gene. Lots of great options. And just a nice example of how we might use this approach to go after other traits that are deleterious for us in the lab. And with that, I would like to thank Maitreya and the Genome Lab, especially my co-authors on this paper, Clara, Erin, Kalina, and Katie. Clara is the one who sent me that great TUP1 result right before my talk. And my funding sources, and a special thanks to the Cold Spring Harbor Yeast course from 2015, who did some of the bulk segregant analysis as part of their curriculum. And I'm happy to take any ideas. I'd love to hear from you and hope to talk to many of you throughout the week. And with that, I will take any questions. Thank you. Yes. I think that exploring not just strains with different ploidy in these experiments, but also I think there's a wonderful reservoir of evolution potential in natural isolates of yeast themselves that might give us different results and reveal some more interesting mechanisms.
All right, in the interest of time, we'll move on. Thank you so much, Elyse.